Welcome to the deep dive. Today we're taking a really close look at the basic tools, the fundamental mechanisms that scientists first used to map the genomes of bacteria and bacteriophages, the viruses that infect them.
Yeah, this is really the bedrock of molecular genetics. Our goal here is to give you a clear path through the core concepts, the key experiments, and, the terminology for genetic analysis, recombination, mapping, all in these simpler organisms.
Think of it as a shortcut, right? To
Yeah.
understanding how we first learned to actually read the genetic instructions in microbes.
Exactly. And why bacteria and phages? Well, they were perfect for early research for a couple of key reasons.
They're fast and simple.
You got it. First, super short reproductive cycles. You can get billions of cells, billions of phages really quickly. That lets you see rare genetic events happening.
Okay.
And second, you can study them in pure culture, just one species or even one specific mutant strain all by itself.
Right. Keeps things clean, experimentally.
Absolutely. That simplicity was crucial for figuring out the basics of gene expression, how genes are regulated, and later on, essential for developing things like recombinant DNA technology cloning.
Okay, so let's lay some groundwork. Basic bacterial genetics. We know bacteria mutate spontaneously, just like more complex organisms. The Luria and Lederberg experiment proved this, yeah. Showing resistance wasn't directed by the environment.
That's the one. And mutations are pretty easy to study because bacteria are usually haploid.
Meaning just one copy of their chromosome.
Correct. So if a gene changes, you see the phenotype right away. There's no second copy masking it.
And we need to know the difference between the main cell types used in these experiments. The wild type is called a prototroph.
Think of proto as first or original. These guys are self-sufficient. They can grow on a minimal medium, just basic salts and a sugar, because they can synthesize all the essential stuff like amino acids, vitamins themselves.
Then there's the mutant version, the auxotroph.
Right. Oxos suggests needing help or growth factors. These have lost the ability to make something essential. So a leu minus auxotroph can't make leucine.
So you have to add leucine to its food, its growth medium, or it won't grow.
Exactly. And this difference is super useful for experiments, lets you select for specific genetic changes.
When we grow bacteria in liquid, they follow a standard pattern. Lag phase, then exponential growth, then they level off in stationary phase. But how do we actually count them accurately? There are billions in there.
Right, you need to quantify. That's where serial dilution and colony counting is essential.
Okay, break that down.
You take your dense culture and you dilute it a lot. You do it in steps maybe 1 to 100, then take some of that and dilute it 1 to 100 again, and so on. Maybe 5 or 6 times.
So you end up with like a 1 in 10 million dilution or something?
Could be, yeah. Then you take a small, known volume of that final dilution, spread it on an agar plate. Each viable bacterium grows into a visible spot, a colony.
You count the colonies.
Count the colonies, multiply back by the total dilution factor, and you get a good estimate of the original cell density. It's a way to make huge numbers countable.
Clever. Okay, which brings us to the really cool part. How bacteria actually share genetic information. They don't have sex like eukaryotes, but they do genetic recombination.
Yeah, 3 main ways: conjugation, transformation, and transduction. And the key thing is it results in an altered genotype. Genes from one cell replace genes in the host cell.
And this isn't just lab stuff, right? This happens in nature.
Absolutely. We need to distinguish vertical gene transfer parent to offspring, same species, from horizontal gene transfer. That's gene exchange between different species, sometimes even distantly related ones.
And that horizontal transfer is a big deal medically.
Huge. It's how antibiotic resistance genes, or genes for pathogenicity, can spread so rapidly through bacterial populations, a major public health concern.
Okay, let's tackle the first method, conjugation. This requires direct cell-to-cell contact.
This was the first one discovered. Lederberg and Tatum back in 1946. They mixed 2 different multiple auxotroph strains. Strain A needed, say, methionine and biotin. Strain B needed threonine and leucine. Neither could grow on minimal medium alone.
But when they mixed them,
They recovered rare prototroph cells that could grow on minimal medium. Only about 1 in 10 million cells, but it proved genetic exchange must have happened.
how do they know it required physical contact?
That was the Davis U-tube experiment. They put the 2 strains in different arms of a U-shaped tube, separated by a filter. The filter let liquid medium pass through, but not the bacterial cells themselves.
No prototrophs appeared.
Nope. Proved that direct physical contact was absolutely necessary for this type of gene transfer.
And that contact happens via something called the F pelus, or sex pelus. What's driving this?
The F factor. Stands for fertility factor. It's a plasmid.
Define plasm again.
It's a small, usually circular, double-stranded DNA molecule that exists separately from the main bacterial chromosome. The F-factor plasmid contains special genes called tray genes, which are needed for making the pilus and transferring the DNA.
So cells with the F factor are donors, F++. Cells without it are recipients, F.
Exactly. And in a typical F plus X F mating, the F plus cell makes a copy of the F factor and transfers it through the pilus to the F cell.
So the F cell becomes F plus tray.
Almost always. But here's the thing. In these standard F plus X F crosses, recombination involving the main chromosomal genes is actually very rare, like that original 1 in 10 million frequency.
But then there were these special Hfr strains, high-frequency recombination.
Right. These strains recombine chromosomal genes about 1,000 times more frequently than standard F plus strains, so maybe 1 in 10,000 instead of 1 in 10 million.
But much better odds. But here's the weird part you mentioned. When an Hfr cell mates with an F cell, the recipient usually stays F. Why doesn't it get the F factor?
Ah, that's the key insight. In an Hfr strain, the F factor isn't floating free as a plasmid anymore. It has actually integrated itself into the main bacterial chromosome.
So it's become part of the big circle of DNA.
Precisely. And this led to the brilliant interrupted mating technique by Woolman and Jacob.
This is the blender experiment, right? Sounds kind of brutal.
Yeah, literally. They mixed an Hfr strain and an F strain, let them start conjugating, and then at specific time points, say 5 minutes, 10 minutes, 15 minutes, they'd stick the culture in a blender.
To physically break the mating pairs apart.
Exactly. The shear forces break the pilus connection. Then they'd test the F recipients to see which genes from the Hfr donor had made it across in that time.
And what did they find?
They found that genes were transferred in a specific order, and the timing was consistent. Gene A always transferred before gene B before gene C, and so on. And the longer they let them conjugate before blending, the more genes got transferred.
Like bees on a string being fed through.
Perfectly put. For example, they might see the resistance gene transfer at 10 minutes, the lac gene for lactose use at around 18 minutes, gal gene at 25 minutes. It established a time map of the E. coli chromosome. Genes were ordered linearly based on transfer time.
And this also proved the chromosome was circular. How?
Because different Hfr strains existed where the F factor had integrated at different locations and in different orientations within the chromosome. So one Hfr strain might transfer genes in the order ADCD, while another might transfer DCBA, and yet another might start somewhere else, like MLK.
So putting all those different linear maps together only made sense if the underlying structure was a circle.
Exactly. The integration point of the F factor determined the origin O and the direction of transfer.
Okay, so why does the F recipient usually stay F in an Hfr cross?
Because the integrated F factor itself is transferred last. Think of the whole chromosome starting to transfer from the origin point within the integrated F factor. To transfer the entire F factor, the entire bacterial chromosome would have to make it across.
Which takes about 100 minutes in E. coli.
Right. And conjugation is usually interrupted naturally or certainly by blending long before that. So the recipient gets a chunk of the HSR chromosome, but usually not the tail end containing most of the F factor genes. So it stays F.
Makes sense. So the F factor is a plasmid, but because it can integrate into the chromosome, it's also called an episome.
Correct. Episome is a term for genetic elements that can exist either autonomously or integrated into the host chromosome. Now, besides the F factor, there are other important plasmids.
Like the R plasmids. You mentioned antibiotic resistance.
Yes, these are critically important medically. R plasmids carry genes that confer resistance to antibiotics. They often have two components, the RTF, or resistance transfer factor.
That handles the conjugation part, like the tree genes on the F factor.
Exactly. And then one or more R determinants. These are the actual genes providing resistance to specific drugs. Tetracycline, ampicillin, streptomycin, sulfonamides, mercury, you name it.
And one R plasmid can carry resistance to multiple drugs.
Often, yes. This is a major reason for the terrifying rise of MDR, multi-drug-resistant bacteria. These plasmids can accumulate multiple R determinants and then spread rapidly via horizontal transfer, conjugation, thanks to the RTF component.
Scary stuff. What about col plasmids?
Less immediately threatening to us, usually. They produce proteins called colicins.
What do they do?
They're toxic to other bacterial strains, even closely related ones that lack the same col plasmid. It's a form of microbial warfare.
Okay, one more state related to the F factor, the F prime state.
Right. This happens when an F factor that was integrated into the chromosome, like in an Hfr cell, excises itself to become an independent plasmid again. But sometimes the excision is sloppy, meaning it accidentally picks up some of the adjacent bacterial chromosomal genes along with it as it loops out. So now you have an F factor plasmid carrying, say, the lac genes from the host chromosome. That's an F plasmid.
And why is that useful?
It's incredibly useful for genetic analysis. When an F cell conjugates with an F cell, it transfers this F plasmid. The recipient now has its own copy of the chromosomal genes plus the copies carried on the F plasmid.
So it's partially deployed for those specific genes.
Exactly. This partially deployed cell is called a merazygote. Merazygotes were essential for early studies of gene regulation, things like figuring out dominance relationships between different alleles of a gene.
Okay, that covers conjugation pretty well. Let's move to the second mechanism, transformation.
Right. No direct cell-to-cell contact needed here. Transformation is when a bacterium takes up small pieces of exogenous DNA just floating around in the environment, maybe from arrest cells.
But the recipient cell has to be ready for it.
Yes, it needs to be in a physiological state called competence, where its cell wall and membrane are permeable to DNA. This can happen naturally in some species, or we can induce it artificially in the lab.
So how does the uptake and integration work?
Okay, visualize this. A fragment of double-stranded DNA binds to receptor sites on the competent cell surface. As it enters, usually one of the DNA strands is degraded by enzymes called nucleases.
So only a single strand gets inside.
Typically, yes. This single strand then finds the homologous region on the host bacterium's chromosome. It aligns, and through recombination enzymes, it actually replaces the equivalent segment on one of the host DNA strands.
Creating a mismatch for a little while.
Exactly. That region is temporarily a heteroduplex. It has one strand from the host and one from the incoming DNA, which might have a different sequence, like a mutant allele versus a wild type.
Then when the cell divides,
one daughter cell inherits the original host DNA sequence, and the other inherits the newly transformed sequence. So one cell is transformed, one isn't.
can this be used for mapping genes like conjugation?
Yes, through cotransformation.
Yeah.
The DNA fragments taken up are relatively small, maybe 10,000 to 20,000 base pairs. If two genes are physically located very close together on the bacterial chromosome, they're likely to be carried on the same piece of exogenous DNA fragment.
So they'll be taken up and integrated together.
Right. The frequency at which two genes are co-transformed is inversely related to the distance between them. If they're always co-transformed, they're very tightly linked. If they're rarely co-transformed, they're farther apart. It gives you a measure of linkage.
Another mapping tool. Cool.
Yeah.
Okay, method 3, transduction. This involves viruses.
Yes, bacteriophages, or just phages viruses that specifically infect bacteria. Transduction is phage-mediated gene transfer.
Let's use the classic example, Phage T4. What's its basic life cycle?
T4 has this really iconic structure, icosahedral head, containing DNA, a tail sheath, tail fibers for attachment. It follows the lytic cycle.
Lytic means it breaks open the cell.
Yep. Step 1. Adsorption, the phage attaches to the bacterial cell wall using its tail fibers. Step 2. Injection, it injects its DNA into the bacterium, like a syringe. The protein coat stays outside.
Then it takes over.
Completely. The phage DNA directs the host cell's machinery to stop making bacterial stuff and start making phage components, DNA, head proteins, tail proteins. It often degrades the host DNA too.
Assembly line for viruses.
Pretty much. New phage particles are assembled inside the cell. It's partly self-assembly, partly enzyme-directed. Finally, the phage directs production of an enzyme, like lysozyme, that breaks down the cell wall from the inside.
Pop goes the bacterium.
And releases maybe 200 new phage particles ready to infect neighboring cells. That's lysis.
How do scientists count these invisible phages?
Using the plaque assay. You mix a dilute phage sample with a large number of host bacteria and spread them onto an agar plate. The bacteria grow into a continuous layer, a lawn.
Okay.
But wherever a single phage particle initially landed, it infects a cell, lasts it, releases 200 new phages, which infect surrounding cells, last them. This cycle repeats.
Creating a hole in the bacterial lawn.
Exactly. You get a clear circular area called a plaque where the bacteria have been destroyed. Each plaque originated from a single phage particle. So you count the plaques, factor in the dilution, and calculate the original phage concentration.
Neat. But you said not all phages immediately destroy their host.
Correct. Some phages, called temperate phages, can enter an alternative pathway called lysogeny.
What happens there?
Instead of replicating and lysoving the cell, the phage DNA integrates itself into the host bacterial chromosome, just like the F factor in an Hfr cell.
So the phage DNA becomes part of the bacterial DNA.
Yes. In this integrated state, it's called a prophage. The bacterium carrying the prophage is said to be lysogenic. It's essentially harboring the virus in a dormant state. The prophage gets replicated along with the bacterial chromosome every time the cell divides.
Is the prophage also an episome then?
Yes, because it can't exist integrated or under certain conditions like UV damage, it can excise itself and enter the lytic cycle. Phages that can only lyse are called virulent phages. Temperate phages can choose lysogeny or lysis.
How was transduction actually discovered? Another clever experiment.
Indeed. Lederberg and Zinder, this time using Salmonella auxotrophs. They used that same Davis U-tube setup with the filter separating two different auxotrophic strains.
But this time they did get prototrophs, even without cell contact.
They did, so it couldn't be conjugation. They reasoned there must be some filterable agent, Fa, carrying genetic information across the filter from one strain to the other.
And that agent was?
A bacteriophage, specifically Phage P22, which was naturally infecting one of their Salmonella strains.
So how does the phage transfer bacterial genes? This is generalized transduction.
Okay, during the theolytic cycle, when new phage particles are being assembled, the phage packaging machinery sometimes makes a mistake. Instead of packing phage DNA into the phage head, it accidentally stuffs in a random piece of the host bacterial chromosome, which was being chopped up anyway.
So you get a phage particle with bacterial DNA inside instead of viral DNA.
Exactly. It's a defective phage in terms of viral replication, but it's still infectious. It can attach to a new bacterium and inject the DNA it's carrying.
And if that DNA is bacterial,
it can then recombine with the new host's chromosome, potentially changing its genotype, for example, transferring a functional gene to an auxotroph, making it a prototroph. That's generalized transduction because any piece of the bacterial chromosome can potentially be transferred.
And can this be used for mapping too?
You bet. Cotransduction frequency, just like cotransformation. Genes that are close together on the bacterial chromosome are more likely to be packaged into the same phage head and transferred together. The higher the cotransduction frequency for two genes, the closer they are linked. Another ruler for the bacterial genome.
Amazing how many ways they figured out to map these things. Okay, this brings us to maybe the most intricate part, fine structure analysis, going inside the gene.
Yeah, this is Seymour Benzer's legendary work with the RI locus of Phage T4 in the 1950s and 60s. Before this, people kind of thought of the gene as an indivisible bead on a string. Benzer showed it had internal structure.
How could he possibly map within a single gene back then? No sequencing yet.
Pure genetic ingenuity using a powerful selection system. He worked with RIM mutants of Phage T4. Wild-type phages, $, can infect and lyse both E. coli strain B and E. coli strain K12.
Right.
But RI mutant phages have a specific defect. They can lyse strain B just fine, but they cannot successfully lyse strain K12.
So K12 is the key. It distinguishes wild type from the RI mutants.
Exactly. K12 lambda was his incredibly sensitive tool. If you plate phages on K12, only the wild-type ones will make plaques. The RI mutants won't grow.
So what was the first surprise?
He collected many independent RI mutants when he co-infected E. coli K12 with two different RI mutants simultaneously. Sometimes the K12 cells did lyse.
Wait, how? Neither mutant could do it alone.
This phenomenon is called complementation. It told Benzer that the R region wasn't one single functional unit. It must consist of at least two different functional units, which he called cistrons, cistron A and cistron B.
Like two different genes needed for the same overall process.
Precisely. If you infect with one mutant that has a broken cistron A but a good cistron B, and another mutant that has a good cistron A but a broken cistron B,
then inside the same cell, one phage provides the working A product, the other provides the working B product, and together they get the job done.
You got it. They complement each other. We now understand a cistron is essentially equivalent to a segment of DNA specifying one polypeptide.
Yeah, okay, that's complementation. How did he map within one cistron?
Through recombination. He'd take two different point mutants that were both within, say, cistron A, so they couldn't complement each other. He would co-infect E. coli B with huge numbers of both mutant phages.
Why strain B?
Because both RII mutants can grow on strain B. This allows them to replicate inside the same cell, and very rarely, a recombination event, a physical crossover, could occur between the two mutation sites within the cistron A DNA.
Creating a chromosome with neither mutation. A wild-type cistron A.
Exactly. And maybe also a double mutant chromosome, but he didn't care about that one. He cared about generating that rare wild-type recombinant.
How rare.
Extremely rare, maybe 1 in a million or less, depending on how close the mutations were. But here's the genius. He then took the progeny phages produced from the E. coli B infection, millions and millions of them, mostly parental mutants, and plated them onto E. coli K.
And only the wild-type recombinants can form plaques on K12.
Precisely. The K12 strain acted as an incredibly selective filter, allowing him to detect and count even exceedingly rare recombination events. The frequency of recombination, number of plaques on K12 divided by total phages plated on B, was proportional to the distance between the two mutation points within the gene.
Wow. So he could map the linear order of mutations inside a single gene. But testing all pairs of mutants must have taken forever.
It would have. So he developed an even faster method, deletion testing. He identified and characterized a set of RI mutants that weren't point mutations, but had actually lost larger, defined segments of the RI region deletion mutations. Many of these deletions overlapped.
How did that help?
He could take an unknown point mutant and quickly map its approximate location. He'd cross the point mutant with each of his standard deletion mutants. If the point mutation site was within the region missing in a particular deletion mutant,
Then there's no corresponding wild-type sequence for it to recombine with.
correct. Recombination cannot produce a wild-type phage if the point mutation lies opposite a deletion that covers that site. So if a cross between a point mutant and deletion mutant #7 yielded no wild-type recombinants on K12, Benzer knew the point mutation must lie within the segment of DNA missing in deletion #7.
A much faster way to narrow down the location.
Hugely faster. Using complementation, recombination tests, and deletion mapping, Benzer ultimately analyzed something like 20,000 RII mutations and mapped over 300 distinct mutable sites within just those two cistrons. He even identified mutational hotspots. It was a tour de force, proving the gene wasn't indivisible long before DNA sequencing existed.
Incredible work.
Okay,
Let's try to wrap this up.
so to recap, we've seen three main ways bacteria exchange genes: conjugation, cell contact, F factor, Hfr mapping; transformation, uptake of free DNA, cotransformation mapping; and transduction, phage transfer cotransduction mapping. And then Benzer's work with phage recombination gave us fine structure mapping within genes.
These techniques were absolutely fundamental. They built the foundation for understanding prokaryotic genomes and really all of molecular genetics that followed.
Couldn't have said it better.
All right, so here's a final thought for you to chew on. We talk about our plasmids and the really serious problem of multi-drug-resistant bacteria spreading through horizontal gene transfer, often via conjugation. Now, thinking about what we learned, especially about the bacchylic cycle of bacteriophages, how might we potentially leverage that destructive cycle? Could phages, which are naturally evolved to target and kill specific bacteria, be used as a medical tool? Could we be entering, as the chapter hints, an age of the phage as a way to combat antibiotic-resistant infections? Something to consider.
Definitely food for thought.
Thank you for joining us for this deep dive into the world of bacterial and phage genetics. We hope it was helpful, and we'll see you next time.